Just a quick pause there to let Ali get the recording started. We want to welcome Kelly Turkington this morning. The topic of today's webinar is disease management, scouting, and risk assessment. And Kelly is a research scientist with Agriculture and Agri-Food Canada. He's done a lot of work in cropping systems and their impact on disease levels, both in cereals and oilseeds. So in a lot of our tighter canola cereal rotations, he's got lots of expertise in terms of understanding the risk that those rotations may present for both types of crops within that rotation. This morning, his focus will be on the major canola diseases. Given the timing, he's going to focus a lot on Sclerotinia because that's the disease that we still have an opportunity to manage for this crop in terms of fungicide applications, and he'll talk about assessing risk and getting the most efficacy possible out of those fungicide applications. But also he will touch on blackleg and clubroot in terms of, in particular, scouting this fall to assess what the levels really are in your fields and how severe the yield penalty might be in some of those fields, and whether you need to make any management changes in the future. We've accessed one CEU credit for both the Certified Crop Advisor program and the CCSC program, and so stay tuned. At the end of Kelly's presentation, we'll provide you with the code that you can submit to access those credits. So with that, I think I will let Ali switch presenters and turn things over to Kelly.
Excellent. Thank you, Derwin, for the introduction. So we'll get started this morning and try and move through this fairly quickly. There's a fairly large number of slides. I've got several co-authors that have kindly contributed some information to the presentation, and I'd especially like to thank Randy Kuchar, who I've worked with quite closely over the last number of years. We're very fortunate to have some excellent colleagues, both in Ag Canada, provincial governments, and with the universities and technical staff. And we're fortunate to have funding from a number of different producer groups in the 3 Prairie provinces and the Canola Council of Canada. And finally, I'd like to thank Clint who approached me to present this topic this morning for you. Now, if we look at plant disease, I think this is a nice starting point. If we look at development of damaging levels of plant disease, you really need a virulent pathogen in sufficient quantity. You need a host that's susceptible and has a reasonable yield potential, and you need a favorable environment. And if these 3 factors interact and are optimal for disease development, you have the production of disease and damaging levels of disease. When one or more of these components is less than ideal as far as disease development, you'll end up with a reduced level of disease. And the thing to keep in mind is that a producer really has the ability to manipulate one or more of these components. And if we look at plant diseases and some of the risk factors, and these apply to all the canola diseases that you'd routinely deal with, and any of the other diseases and the other crops that you grow. Probably one of the key ones is rotation. That's probably one of the mother of all risk factors. The other main one that is conducive weather, but if we look at rotation, really tight rotation. So in general, a single year of a non-host crop between the crop that you're interested in is typically not long enough as far as seeing the benefit, the maximum benefit from crop rotation. So you need to be looking at at least a 2-year break between host crops to start to see a large impact as far as rotation. Seed quality can be a factor, and in many cases, depending on the crop that you're growing and the disease, if there's seed-borne disease, it will have an effect as far as seed germination, seedling growth, and stand establishment. So if you have seed-borne disease, that can increase your risk of early season issues, and it can also increase your risk that you may be introducing something into your field that could become a problem several years down the road, and especially if you're in a tight rotation. History of disease, that's a bit of a no-brainer if you've had issues in the past. And when I say issues in the past, you really need to have significant issues. If you've had trace levels of Sclerotinia in the past, that to me isn't a significant or an indication of a significant disease risk. It means that it's there, but likely not a significant risk. Field location, and it sort of relates to history of disease, basically what's happening in adjacent fields. And it's the adjacent fields that are of most concern for things like Sclerotinia, for things like blackleg, and even we're starting to see for things like clubroot and some of the work that I have the fortune of participating in with the U of A, where we're seeing dispersal of the clubroot pathogen via resting spores in wind-dispersed soil particles, so via soil erosion. So field location can be a big problem and risk factor as far as whether you have problems with things like Sclerotinia, blackleg, or perhaps potential introduction of clubroot. Conservation tillage, I've got a big question mark there. We've done a lot of work on this, and if you look at research in the prairies, you look at research in central Canada and elsewhere, really crop rotation and weather are probably the two biggest factors of influencing disease risk. And if you look at conservation tillage and root diseases, we actually see a benefit of moving to a conservation tillage system. And if you look at something like Sclerotinia, actually some of the work coming out of Brazil and the States, mainly with soybean and other sort of grain legume crops, conservation tillage actually has a beneficial effect. Conducive weather, frequent showers, so you need, not rain continually, but showers every couple of days, and heavy dews, the crop canopy staying wet till well into mid-afternoon, and then the temperatures, you really need daily minimum temperatures of greater than 10 degrees C, and maximum temperatures that are less than about 20 to 30 degrees C. Once you start getting into maximum temperatures during the day of 30 or more, that tends to shut down a lot of disease issues. Susceptible variety, again, a no-brainer there. If it doesn't have resistance, it's going to have an issue with potential with a particular disease problem. Lack of balanced fertility. Here, deficiencies can predispose the crop to attack. So in canola, if we look at alternaria black spot, a sulfur deficiency can actually increase your risk that black spot will become more of an issue. Hasn't really been a huge issue in Argentine canola, more of an issue traditionally in Polish canola, but with the tight rotations that we have, it may be something that is becoming more of an issue, and making sure your sulfur fertility program is up to snuff is probably going to help. Excess fertility is also an issue, and especially for something like Sclerotinia. So if you're pushing for maximum yields, pushing on the nitrogen, getting a very dense crop canopy that's more susceptible to plant diseases, basically because you're creating a more conducive microenvironment in that crop. Now, if we look at management strategies, and I'm going to go through these fairly quickly because you can go back to the presentation later on, but really if you think about it and think of disease as being the interaction of a pathogen, a host, and the environment, really the producer's manipulating one or more of these components of the disease triangle. And our philosophy here at Lacombe, and working with my colleagues like Trish Clayton and John O'Donovan and Neil Harker, others like Eric Johnson and Bob Blackshaw and so on, is an integrated approach to crop and pest management. So utilizing as many of the tools as possible to manipulate as many of the components of the disease triangle. So if we look at the host, you're looking at growing a crop with resistance, using balanced fertility, and using generally good agronomics to limit stress on that crop. Any stress that you impose, deep seeding for instance, will increase the risk of seed and seedling issues and stand establishment problems. The pathogen, you can target the pathogen directly using crop rotation, by using healthy seeds, so careful seed choice. Field location can be a management strategy in terms of, if you know that you've had a blackleg problem, for instance, in one field, you may want to avoid planting immediately adjacent to that field. And last but not least, of course, with the pathogen, you can target it with fungicides, whether they're foliar products put in crop or applied to the seed. Finally, the environment, and this is more of a difficult one to manipulate, but if you're under irrigation in southern Alberta, the Outlook Irrigation District, you can look at manipulating your irrigation system, maybe cutting back on irrigation during critical times for disease risk, but again, making sure you're meeting the water needs of the crop, and that may have a benefit as far as lowering your risk. There's been a lot of interest in terms of manipulating the crop, maybe going wider row spacings in terms of Sclerotinia. My feeling there, if you look at the literature and some of our experiences, is that you probably need to go to some very, very wide row spacing. So we're talking probably at least 24 to 30, if not larger row spacings to perhaps see any impact. The row spacings that we typically use that might range anywhere from 7 to 12 to maybe even 18-inch row spacings may not have a huge impact as far as crop canopy in terms of Sclerotinia management. Now, if we look at the presence of disease, and this is something that I often get asked to go out and look at fields in mid to late summer or even early fall, and in some cases the crop scout or consultant or producer on the phone are quite concerned about the disease that they have, and you get out to the field and you have a look at it and you say, yeah, you can find some Sclerotinia here or maybe a bit of blackleg, but really the level that you have there is not causing a significant yield loss. So you really need to put it in perspective. Is it a significant amount? If it's trace or low levels of disease, so if it's less than about 10% infected plants with Sclerotinia, you're probably going to have a limited impact on crop productivity and quality. But if you start to see those levels of Sclerotinia or blackleg creeping up, so you're looking at more than about 10 to 20% infected plants, the severity of symptoms that you have are starting to have an impact, so the large cankers, let's say, at the base of the plant as far as blackleg, or large spreading lesions in terms of Sclerotinia, and they're affecting the main stems or the main branches, it's going to have a significant impact on yield and kernel weights and grade in the crop that you're looking at. In terms of Sclerotinia, probably the key time, if you're wanting to assess the sort of end result of it or the ultimate impact, that's done best between flowering, the end of flowering and swathing. So as the crop, just before the crop starts to turn, that's you're going to give your maximum level of disease and a very good level of expression. So if you go out to the field and you assess in the field, probably at 5 to 10 locations, the percent of infected plants, and you're focusing mainly on infections that are affecting the main stem or main branches, you get that number, the percent infected plants, you then multiply that by 0.4 to 0.5. That gives you an approximate indication of the percent yield loss. So if you're looking at a 10 to 20% level of incidence in terms of main stem or main branch infections, your potential yield loss is in that 5 to 10% range. And that's roughly a threshold that we see as far as the need for fungicide or not spraying. So of course, as the price of canola increases, that threshold becomes lower. So if canola is $6 per bushel, you're probably wanting to see at least 20% infected plants in that field to perhaps have an economic benefit from that fungicide. Now, if we're going to focus primarily on Sclerotinia, but I'll bring in blackleg and clubroot in the presentation. But if we look at Sclerotinia, it's caused by the fungus Sclerotinia sclerotiorum, and if you look at affected plants in the field, they will, and especially if the affected tissues are starting to dry down, so the plants are starting to mature, that tissue is extremely brittle. It shreds and shatters very easily, so it's quite characteristic compared to yellowing that might occur due to drought stress, or you might have some branches on a plant that just die for no apparent reason, or perhaps you're looking at other issues like clubroot or a blackleg, or maybe even fusarium wilt. Typically, you don't get this very brittle and very easily shredded and shattered stem tissue. So if you take that stem and move it back and forth in your hands, you'll see that it shreds and shatters very easily. Typically, the affected tissue has this bleached appearance, which can be range in color from a sort of a whitish to maybe even a light tan or a light grayish color in appearance. Sometimes you'll see evidence of fungal growth on the outside of the plant, and even occasionally sclerotia, which are the resting bodies of the fungus. But if you cut that affected stem in half, you'll often find sclerotia within the affected tissues. So very quickly, we'll just go through the disease cycle of Sclerotinia. Again, it's important to know the enemy that you're dealing with and to understand it in order to make sure that you're using the most appropriate strategies to combat it. So if we look at Sclerotinia, it survives in the soil as these resting bodies called sclerotia. An individual one is called a sclerotium. Cut it in half, it's got this black outer rind and white interior. Basically a very compact mass of hyphae that function as a survival structure for the fungus. And they can survive in the field anywhere from 2 to 3 years perhaps. By that time, a lot of them will have decomposed, but they're fairly long-lived. In mid-June, once the crop canopy starts to cover the soil surface, and that can be a canola canopy, it could be a cereal canopy, it could be a pulse canopy, you're going to have moisture that comes into the field via rain being held in the crop canopy. You're going to maintain the soil close to field capacity, and these sclerotia will start to germinate and produce a little stalk. Once that stalk emerges out of the soil, light will trigger the formation of this disc or apothecium, and if you look at it inside of it, it looks like a golf tee. So you need moisture at or near field capacity and a moderate temperatures in that range of 10 to 20. If you start getting much cooler than 10 degrees C, then this is work done on average temperatures. But if you're getting very cool conditions or very warm conditions, those are typically not conditions that favor germination of these resting bodies. In terms of dispersal of the pathogen from these little apothecia or mushroom-like structures, fungus actually shoots spores up into the air like little cannonballs. So you get thousands, if not millions, of spores being shot up into the air. Basically, production of these spores occurs over quite a wide range, 4 to 32 degrees C. If you look at the literature, you need fairly wet conditions, greater than about 98% relative humidity, and the optimum temperatures in that 22 degrees C range. In terms of dispersal, if you look at all the work that's been done, it's not going to be tens of kilometers. It's really probably maybe tens to hundreds of meters or yards. So it's really adjacent fields that are most at risk from this disease. So most of the spores will be dispersed and will fall back down to the ground within about 100 to 200 to 300 meters or roughly yards from the source. One thing to keep in mind is that if you get a lot of rain, so it rains constantly, you'll get water droplets that form on the top of these apothecia and actually prevent the fungus from releasing spores into the air. Excessive rain will wash the spores out of the air, so if you get constant rainfall, that's actually not very conducive to Sclerotinia. What you need is several days of wet conditions. You get germination of the resting bodies, which occurs over probably a 2 to 4 week period, depending on conditions. Production of the apothecia, and then if you get a couple days where it's fairly windy and it's not raining, those are ideal conditions for dispersal of the pathogen. So it'll move up onto the inflorescence of the canola plant, so it'll land on the canola petals. If you look at that under the microscope, here you can see some spores of the Sclerotinia fungus on petal tissue. The spores will start to germinate and produce germ tubes and hyphae. And the fungus needs the petal tissue as a carbon and nitrogen source in order to infect the plant. So if these spores land on a leaf or a stem, they don't have enough energy reserves within themselves to directly infect the plant. So the petals are a key component of the disease cycle of Sclerotinia, and that's why some of the forecasting systems actually focus on the petals. So in terms of infection of the plant, really it can occur over a range of about 10 to 30 degrees C, optimum in that 20 to 25 degrees C range. You need moisture, either free moisture in terms of a layer of moisture on the plant tissue, or relative humidity is close to 100%. An infection from spore landing to germinating, a petal lodging in the leaf axils and leaf bases, probably infection from the spore to the petal to the plant. Once those petals, of course, have fallen out of the flower and lodged in leaf axils and bases, it can take probably anywhere from 48 to 72 hours, depending on conditions. So here you can see some canola leaf bases and axils where you've got clumps of petals that collect, and those will hold droplets of moisture, so it's an ideal environment for the fungus. So if you're looking at fungicides in terms of managing Sclerotinia, often you'll see comments made that you want to get good petal coverage. My philosophy is, yes, perhaps, but the petals are in the inflorescence or in the flowers for about a 6-day period, then you get a new crop of petals produced. If you look at a crop that's flowering for 2 to 3 weeks, depending on conditions, you've got a number of different crops of petals being produced and potentially infested with Sclerotinia. So if your main focus was on spraying and protecting the petals, you'd have to go in and spray the crop several times during the flowering period. So in addition to the petals, you want to make sure that you use good water volumes, good spray pressure, because you want to move that chemical not only onto the petals, but into the crop canopy, onto the leaf axils and leaf bases, where the actual infection is going to occur. So once you get towards swathing, the end of flowering towards swathing, that's when you typically start to see symptoms of Sclerotinia, and you can be driving by a field and see the odd plant or many plants actually showing signs of premature ripening. There's some good examples of that here. Don't assume it's Sclerotinia. It could be some of the other issues. It could be blackleg, and here's a couple of pictures from Richard Google, a colleague in Saskatoon, showing you some lower stem lesions, basal stem canker. Typically, you get a light-colored interior to the lesion, can be whitish or tanny in color. You get a dark purpley black margin, and the lesions are dotted with little fruiting bodies of the blackleg fungus. Sometimes, if you've got wet conditions, the fungus will actually start to ooze out the rain splashed stage of the blackleg fungus, and this reddish or pinkish material is actually the rain splash stage. So these are very characteristic symptoms of blackleg. Another example of a lower stem lesion or a lower stem canker, you see cankering and cracking of the stem, purpley brown margin, and it's dotted with these little dots that are the rain splash stage of the fungus. Blackleg can also affect the crop at the seedling stage. That's not the case with Sclerotinia. If you see symptoms in Sclerotinia, it will only be as the crop is getting in probably to full bloom or into late bloom and into that period between the end of flowering and swathing. Blackleg, you can see the symptoms right from the seedling stage through to symptoms on the pods, actually, or even at the rosette stage. And here's a couple pictures of very severe infections, likely some variety West Star, probably. These pictures are from Saskatoon. Here's some more current pictures from Randy Kuchar showing you a range of symptoms on more recent canola varieties. Sometimes the symptoms in the newer varieties aren't quite as characteristic or similar to what we would have seen in the past on West Star. You can get some cankering, but often you get blackish discoloration, some evidence of some dots, which is the rain splash stage. You can see that here. But if you cut the stems in half, you'll see extensive discoloration if you've got heavy infection moving into the interior of that stem. So it's affecting the plant's ability to take up and transport water and nutrients, and that's what ultimately causes issues with with yield loss and green filling. You can also see, in some cases, patches. If you see very distinct patches in the field and they're associated with lower spots, and they tend to have more of a yellowy appearance rather than sort of a bleached white appearance, you're probably looking at an issue like clubroot. So if you go into the crop and you see symptoms like that, always, no matter whether it's canola or cereals, dig the plants up. Have a look at what's happening to that root system. Here's a nice big ugly gall of clubroot. And again, clubroot, mainly you're going to see that as the crop is getting into flower and past flowering, but you can also see if it's a very severe infection and there's heavy levels of resting spores in the soil, symptoms quite early in the season, even at maybe the rosette to early flower stage. And typically here you're seeing some wilting because the root system is compromised in that canola plant, can't take up enough water and nutrients to meet the needs of the above ground part of the plant, and as a consequence it starts to wilt and yellow. So if you look at Sclerotinia, here's some very typical symptoms. Typical infection here would have probably resulted from petals that lodged in the axis of these two branches, or where this leaf was attached, moved from the petals into the stem tissue and up and down the plant. Or you can get very extensive development. And this is in a crop in the Leduc area a few years ago where there's a fair bit of lodging. So you had maybe infected stems laying against uninfected stems, and the fungus will readily move from an infected stem to a non-infected stem. So lodging can increase your level of Sclerotinia. And it's a bit of a chicken and egg scenario, which came first. So very quickly, if we look at Sclerotinia, it's a very interesting disease in that if you look at this data, it's old data, but it's still relevant. It's from Morel and Duchek, and it looks at years since previous rapeseed crop and the number of fields with greater than 5% infected plants. So as you increase your number of fields with greater than 5% infected plants, you would expect that under the tighter rotations you'd see more Sclerotinia. And there really is no relationship here between crop rotation and the level of Sclerotinia. More recently, if you look at the mid-90s, there was quite a large Western Canadian canola disease survey. Here it looked at years since previous canola crop and years since previous Sclerotinia susceptible crop. You can see there's no relationship whatsoever there. It doesn't matter whether you're in a one-year rotation or a 5-year rotation, your level of Sclerotinia is fairly similar. If you compare that or contrast that with blackleg, this is some work from Randy Kuchar at Scott and Melfort, you can see with susceptible variety, as you extend the rotation, so this is continuous canola, a canola wheat rotation, a canola wheat pea canola rotation, and then a wheat pea wheat canola rotation, you can see consistent reduction in both the susceptible and the resistant varieties, consistent reduction in blackleg. So rotation has an important role to play, and that's also the case with clubroot. With clubroot, though, unfortunately, you're looking at much longer rotation. Well, why do we see this lack of a relationship with Sclerotinia? And here are a couple of characters here that I had the pleasure of spending some time out in some fields a number of years ago now in sort of Leduc to Sedgwick area. We're looking at some south thistle and Canada thistle, I think, that's infected with Sclerotinia. Sclerotinia has a huge host range. All the broadleaf crops that you typically grow, so field peas, lentils, chickpeas, and so on. So these crops can also support the disease. The other thing is that it is dispersed from one field to the next. So you may have an excellent rotation in your field, and a neighbor who's had a field pea crop or canola crop and a lot of Sclerotinia, that's going to act as a source of disease. Seeding date and Sclerotinia. I'm going to go through this very quickly to try and stay on time and get you done at a reasonable time. This is just some work we did back in 1998 looking at fall seeded canola. So we had fall seeded or dormant seeding. We had very early spring seeding, so usually about the 3rd week of April, roughly, I think was the earliest that we went in, and then usually around the 5th to maybe the 15th of May, so our normal spring seeding date. We looked at the percent infected plants with Sclerotinia, and we had a slightly higher level of Sclerotinia with the fall seeding date and the April seeding date versus the May seeding date. We looked at the results in 1999, a complete reversal in trend and a much more dramatic change. So our fall seeding had much lower levels of Sclerotinia, intermediate with our sort of early spring seeding, and very high levels with our normal spring seeding. So in terms of seeding date, whether it's early or late in the spring, it's probably not going to have a huge impact on your level of Sclerotinia. Really what's happening is, the key thing is, when is that moisture coming into that field, and does that moisture coincide with the crop coming into sort of the rosette stage of development to hold the moisture in to promote germination of sclerotia, and do you get moisture occurring during the flowering period? Those are sort of the key things. Seeding rate. There's some variable results here. We've done work over the years. Here's some work from Lacombe showing no relationship between seeding rate and Sclerotinia. But if we look at some work from Clint Yerke with the Canola Council as part of his master's thesis here at Carmen in 2002, Clint looked at a range of seeding rates, anywhere from 2.2 kilograms per hectare up to 20 kilograms per hectare, and a number of different varieties. You can see this apetalous variety, highlight 201, had the lowest level of disease, some of the other varieties here. But you can see it was really only once we got to that really ultra-high roast seeding rate that we saw a significant increase in Sclerotinia. So seeding rate can have an impact. It's probably where you're pushing your seeding rate to the extreme. If you look at this yellow bar here and all of these graphs, that's sort of the normal seeding rate, 6.7 kilograms per hectare, and not a lot of difference in terms of comparing that to a lower seeding rate of 2.2 kilograms per hectare. So if you look at talking to Randy, Debbie McLaren, or myself, we work on Sclerotinia. It is a tough disease to work on, and we certainly do not pretend that we know all the answers to it. It's a difficult disease to work on as far as doing fungicide trials or even some of the integrated crop management trials that we've done. Some of the factors, or if you look at Sclerotinia as far as a disease, it has the potential to cause substantial losses. It's variable in occurrence from crop region to year. It's got an extensive host range. It affects a range of broadleaf weeds and crops. The yield loss is variable depending on when the fungus comes in terms of the crop, the growth stage. Does it coincide with moisture? What the yield potential is? What's the percentage of infected plants? Is the infection largely confined to the pods versus the stems, the lower stems? If it's mainly the lower stems, you're going to have a much more significant impact on disease. If that infection has come in towards the latter part of the full bloom stage or into late bloom, chances are it's not going to affect mostly the upper part of the crop canopy, and you're not going to have a significant impact on yield. And ultimately also the weather will have an impact. The successes of cultural methods of control like rotation is limited, and I've mentioned that already. The longevity of the sclerotia, 2 to 3 years at least, wide distribution, and the potential for it to move from one field to another. This variability that we see really makes routine fungicide application questionable in a number of areas of Western Canada. If you look at some areas like the Red River Valley, where you're getting more consistent moisture conditions, or maybe in the Barhead, Westlock, Fort Saskatchewan area around Edmonton, can be quite wet, you're going to have Sclerotinia challenges probably more frequently. Some of the factors related to Sclerotinia, and again I'm going to go through this fairly quickly. This is some work from this survey back in the mid-90s looking at nitrogen rate. Basically, as you're increasing your nitrogen rate, you're just simply promoting a more denser crop canopy, and that results in a more favorable microenvironment for disease. So higher levels of disease with higher levels of nitrogen. And we see that in some of the work that we've done here at Lacombe. This is 50% of soil test nitrogen, 100% of soil test nitrogen, and 150%. So we double our level of Sclerotinia by going from 50% of soil test nitrogen to 150%. Lodging. This is, again, some crop specialists with the province of Alberta. When they had specialists back in the late 90s, we were in the Olds area looking at some fields. And this was a field of canola that was laying flat. It had a heavy level of infection and very heavily lodged. So that, again, which came first, the lodging or the Sclerotinia? If you have a variety that's more prone to lodging, it's going to increase your risk of Sclerotinia as more of an issue. And if you have a crop that lodges because it's more prone, the variety, or because of other conditions, you're going to have a greater spread of Sclerotinia. If we look at lodging, as you increase your seeding rate, Clint found in his master's thesis, again, where you're going to those very extreme seeding rates, so 13 to 20 kilograms per hectare, you're going to see a big increase in the amount of lodging. Not so much if you're going from that 2.2 to 6.7 kilograms per hectare. One of the other interesting things that Clint found was the correlation between disease levels and the number of petals on leaves. And if you looked at all 11 cultivars that he tested, there was quite a significant and fairly tight rotation with the number of petals on leaves, and the level of disease. So basically, as the number of petals on the leaves increased, the amount of disease increased. And this is not surprising given the nature and the importance of the petals in the disease cycle. If we look at only the petal varieties, the relationship wasn't as tight and wasn't significant. So probably the main difference here is you've got the apetalous canola variety, so they don't produce any petals, and as a consequence, they don't have as much disease. So that's something, a technology that we don't currently have access to right now, but it can have an impact on Sclerotinia. So let's get into looking at assessing the risk. And if you look at Sclerotinia, my philosophy here is you need to look at all three components of the disease triangle. The host, the pathogen, and the environment all have a significant impact on the risk of Sclerotinia, so you need to account for all three when you're looking at assessing risk. And they're roughly equivalent, but probably the environment holds the trump card as far as Sclerotinia. If your maximum daily temperatures are in that 30 to 35 degrees C range and the next 2 to 3 weeks are forecast to be dry, you're not going to have disease no matter whether you have a susceptible host and the pathogen there is there as far as lots of apothecia and so on. The reason for this is basically that Sclerotinia is a monocyclic disease. So you get sclerotia overwintering in the soil. In mid-June, they start to germinate to produce the apothecia. Those produce spores and release them. And those land up in the petals. Those petals fall off, lodge on leaf axils and bases. The fungus uses the petals as a carbon and nitrogen source, goes into the plant, producing disease, and ultimately a new crop of sclerotia. So the amount of disease at the end of the growing season is proportional to the amount of inoculum floating around in the air, so the spores in the air in June, late June and July, and to a certain extent the number of apothecia that you have being produced. Of course, this relationship is also modified by the environment. So the more favorable the environment is, the more disease and the yield potential of the crop. In contrast, if you look at net blotch and barley, here the host and the environment are the two primary factors, less so the pathogen. And the reason for this is that it's a polycyclic disease. So there's many cycles of spore production and host infection during the growing season. So you have symptoms of leaf disease when you're scouting for weeds in June. Those lesions are producing spores. Those spores are dispersed to adjacent plants or from lower leaves to upper leaves. They germinate to produce a new crop of lesions, which produce a new crop of spores. And this life cycle can complete itself within a week. So that disease will continually develop throughout the growing season. So it could start from a fairly low level in late May, early June, to become quite a significant level as the crop's coming into flag leaf or head emergence. Late blight of potatoes and ascocteta blight of chickpeas are sort of the extreme. Here, the environment is the overriding factor. It's the primary factor driving epidemics of potato late blight and ascoctitis. So you can look at a forecasting system based solely on weather conditions for potato late blight, and it's been very effective as far as assisting potato producers in terms of initiating their spray programs and then how frequently they put on fungicide in their crop during the growing season. And I won't go into the disease cycle here, but again, potato late blight, the fungus that causes it, Phytophthora infestans, is a polycyclic disease, and it cycles very, very rapidly on the potato crop. If we're looking at disease risk, a useful tool that you can look at is the checklist. And this is from the Canola Growers Manual. You can look at a number of factors in terms of the number of years since the last canola crop, disease incidence in the last canola crop. So these are looking at the pathogen and the potential for the pathogen to be there at a level that's going to cause a concern, the crop canopy density, so what effect the host has, and then ultimately the rainfall and the weather as far as potential for infection, and also the risk for apothecia development in the region. And if you total your point values, so you look at this, you say, okay, I've only got 1 to 2 years since the last canola crop, so my risk point value is 10, and you go on and answer questions and assign a point value. If the total point value you have for all of the questions is more than about 40, likely spraying is going to be worthwhile. Now, I just want to take you through, we're getting towards probably the last third of the presentation, just sort of my philosophy as far as assessing risk of Sclerotinia. I worked on using petal testing in the mid-80s, the mid to late 80s, and we're doing some more recent work on this in terms of forecasting risk. But I've worked with people like Derwin and others in the Canola Council and producers and crop scouts over the years and our own experiences, and I just want to try and pass this on to you as far as coming up with a reasonable assessment of risk. Let's look at the host first of all. So when does canopy closure occur, and do you have rainfall occurring at that stage? So that's going to promote germination of the sclerotia and production of spores. When the crop is getting into flowering and the period of flowering, does that coincide with favorable weather conditions which favor production of apothecia? You have spores being produced in the air, they land on petals, and then the conditions, do you have enough moisture there, are the temperatures reasonable as far as favoring disease? What's the lodging potential of the crop? If it's a more lodging-prone variety, that increases your risk of Sclerotinia. What's the yield potential? And really, if you've got a yield potential in that crop that's much less than about 25 to 30 bushels per acre, you're likely not at a high risk of Sclerotinia. You're likely not to see a response from fungicide application. It's really only once you start to see yield potentials in the 30 to 40, 50, 60, 70 bushel per acre range, and perhaps even higher. And it's going to be influenced by, to a certain extent, the variety, but primarily the fertility regime and especially the nitrogen rates that you have. So the more yield potential you have there, the greater the crop canopy density, the more favorable the moisture conditions you have, and the more disease. And last but not least, as far as the host factors, or host-related factors, commodity price. And the higher the price, that threshold as far as spraying becomes much lower. Now, if we look at the pathogen, there's a few things that you can look at here. I spent a lot of time hunting around on my hands and knees looking for apothecia, but if you can look at history of Sclerotinia in your field or area, and not just anecdotal reports, but you have had significant levels of Sclerotinia. When I say significant, at least 20% infected plants, and we're dealing with mainly with lower stem or main branch infections. Do you find apothecia in the fields? And you can look in the field itself, and it's also important to look in adjacent fields. Are they easy to find within 20 to 30 minutes? If you can find them easily, to me that sends a signal that there's an elevated risk of Sclerotinia. If you're in there for 1 or 2 hours, you may have the opportunity to spend that time in the field. It doesn't mean that there's no risk, but certainly the risk is probably lower. You can look at testing petals, and Discovery Seed Labs provides a kit to do this, and we're doing some other work right now looking at using molecular techniques for this. But if you have more than about 30 to 45% petal infestation as the crop's coming into early and into full bloom, that level of petal infestation is really probably a level where you're going to see a significant risk of Sclerotinia. Obviously, the risk increases as the level of petal infestation increases. The thing to keep in mind is this can change over the flowering period. So you can go from very low levels of petal infestation, so essentially no spores in the air as the crop is coming into early bloom, to a situation as the crop's getting into full bloom, you have lots of spores floating around in the air, and you may have petal infestation levels in that 50 to 60 to 70% range. So you have an increased risk when these factors here all coincide with frequent showers, unsettled conditions, and increased yield potential. There's a limited risk even if you have these factors, if the current conditions are dry and temperatures, maximum daily temperatures are forecast to be 30 degrees C and plus. And so if you go out into the crop canopy or into the crop and the canopy is dry at 8 or 9 in the morning, the risk for Sclerotinia is going to be fairly low. Now, if we're looking at apothecia, just to touch on this because there's lots of confusion regarding this, and this is a picture taken from the mid-1980s, and here's the speaker, Kelly. I did have hair at that time, and my midsection is a little slimmer, but you can actually see who was doing most of the work during the summer. This is my supervisor, Robin Morrell, and one of the other summer students. Actually, I shouldn't say that. We came in and they brought new rain suits for us. So I'd been out but we basically crawled around on our hands and knees looking for apothecia as part of Robin's fungicide spray trials that he was setting up in commercial fields. So if you look at Sclerotinia and apothecia, if you look at these two structures here, these are typical apothecia of the Sclerotinia fungus. This is a more immature one, more of a mature one. Here's a good example here. If you follow that back, the stipe, eventually it connects back to a sclerotium. Here's a couple more in the soil. So they're being produced from these resting bodies either on the soil surface or just below the soil surface. You can't see a lot of these other structures. Here's the bird's nest fungus. That is a fungus that decomposes canola residue. And the reason why it's called a bird's nest is it actually has these spore-producing structures that it almost appear like eggs within a bird's nest. So they're called the bird's nest fungus. I've seen spray decisions based on the presence of these structures, and this is not Sclerotinia. The other thing that we've seen very frequently is spray decisions made on the presence of these little tiny structures that are apothecia, but they're not the Sclerotinia fungus. If you compare the relative size of the two, they're quite a bit different. These are very, very small, maybe a millimeter or so in diameter, versus maybe 5 to 10 to maybe 15 millimeters in diameter for these apothecia of Sclerotinia. And this just shows you again a close-up of these structures. I've been in fields where spray decisions have been made based on the presence of these structures, and where we've spent a couple hours in a field and we're unable to find any apothecia of the Sclerotinia fungus. Now, if we look at one of the other very useful tools that is out there is the Canola Council Sclerotinia card, and it's very useful in terms of identifying Sclerotinia as well as the apothecia, and then also has, I think, the checklist as far as assessing risk. So if you don't have one of these, certainly get a hold of one. You can do petal testing, and this just slide shows you an agar plate. So basically the test consists of plating out canola petals on an agar medium, and then a variety of different fungi grow out, and based on the appearance of the particular fungus, you can identify it as being Sclerotinia or something else. So this large colony of fungus here is the Sclerotinia fungus. These dark-colored ones here, orangey, are not. So if you look at the work that we're doing recently, most recently on petal infestation, and one of the issues with using agar plates is it takes about 5 days to do the test, whereas using some of the molecular techniques or modern molecular techniques, that turnaround time can be as short as 1 day. So we're looking at a quantitative molecular test for detection of Sclerotinia on infested petals or flowers, and this work's being done at the U of A with collaboration across the Prairie region with funding from the Canola Council and the Canola Cluster. So this is, I think, a technology that I think will be quite useful, and it's a technology that can be readily deployed to seed testing labs. And if you look at the clubroot test, for instance, that's being done, especially the molecular test, being done routinely by seed testing labs in Alberta primarily, but perhaps also elsewhere. The environment, last but not least. If you look at June, you've got moisture in June, you've got rainfall occurring to maintain soil near field capacity, and this is especially important under a well-developed crop canopy, be it canola, field peas, cereals, whatever. So you're basically promoting germination of the sclerotia. And then as the crop is coming into flowering, you need frequent showers and moderate temperatures. So rainfall of at least 5 to 10 millimeters on more than about 2 days per week. You need humid conditions within the crop canopy, because that will actually tend to allow the petals to stick to the plant. If it's completely dry in that crop canopy, those petals that lodge and fall off do not stick to the plant and thus can't contribute to the infection. Is the crop canopy wet throughout most of the day? So basically, if you go out to that field and it's 2, 3, 4 in the afternoon and your pants are wet up to your knees or higher, that would, to me, would signal that the moisture is persisting in that canopy for a long enough period of time that it's going to be a significant factor as far as elevating the risk of Sclerotinia. If you go into that field at 8 or 9 in the morning and it's bone dry on the soil surface, the crop canopy is bone dry, those conditions are not conducive to infection. Irrigation, I've got a question mark there. I won't go into a lot. Irrigation can be a big factor in terms of some of the bean crops. Canola, irrigation, if you look at an irrigation regime, you're putting on moisture. So prior to the moisture coming into the crop from the irrigation system, it's going to be probably warm and dry. You get a spike of moisture in the crop, and then it tends to dry out fairly quickly. However, if you're looking at a very heavy crop, that irrigation could add moisture there, and that crop canopy could hold that moisture and perhaps increase your risk of Sclerotinia. What about fungicide application? So really, and we're getting towards the end of the presentation now, so we want to assess disease risk and determine whether you need to apply a fungicide or not. And with Sclerotinia, you need to apply the fungicide prior to the appearance of symptoms in the crop. So if you're going out into the crop and maybe the client that you're working with, or if you're a producer yourself, and you come back to the crop and it's already in full bloom at least, and you go out to the crop and you start to see some early evidence of symptoms, it actually is probably too late to spray. You really need to put that fungicide on prior to the appearance of symptoms on the leaves and stems. We also want to ensure good coverage, and I've touched on this already. Don't just focus on the petals. You want to try and get that chemical into the crop canopy, into those leaf axils and bases, where it's going to provide some additional protection as far as preventing infection. Increase your water volumes, and again, just helps with pumping chemical into the crop canopy. Split applications can be quite useful. If you're in that 50 to 70 bushel per acre range, you're in the Red River Valley, you're in maybe that Edmonton area, or an area where you've got a lot of moisture, and you've got a very high disease pressure, you may want to go in twice in that crop. And but the split applications are useful because they allow you to fine-tune your application depending on the identified risk. So if you're not sure that you've got a huge risk, you can go in with a reduced rate of chemical depending on the product that you're using. Some of them are registered for a range of rates. Put the product on, then you can reevaluate the risk. And if the risk decreases, the temperature increases, and there's no rainfall, you can say, okay, the risk is such that I don't need to go back into that crop. The other thing that to keep in mind, and something that I wasn't aware of, but is if you've got a crop where you had variable emergence occurring due to maybe dry conditions initially, and then you get wet conditions at the time of seeding and seedling growth, so you get a variable maturity in the crop. Here, a split application might be useful as far as providing a wider window of protection in that crop canopy. We can talk a bit about fungicide application. This is some older data from Randy Kuchar and Tom Wolf. Here's Sclerotinia incidence. Here's the unsprayed running around 50% infection. Ronalan, which is no longer registered, I think, around about probably 3% infection. Phenolate was probably around 7%. And you can see the yield benefit here. So we've got about a 30 to 33% yield increase by putting on a fungicide. So that was 1998. 1999, basically, they found that they had no significant impact of fungicide application. So they didn't have any real huge benefit as far as yield increase between the untreated or whether it was sprayed with Ronalan or Benomyl. If you look at more recent data in North Dakota, a fellow named Bradley looked at a range of different fungicides, including some of the products that are currently registered. In canola, he looked at a range of timings and basically found that a number of these products could be used to reduce Sclerotinia levels, but that in some cases yield increases did not always result from the application of these particular products. And he suggested that it was associated with the different dates of application that occurred and when the crop was, at what crop growth stage the crop was at, what bloom stage. So what he was suggesting is that you may need to also consider factors in addition to just simply the crop growth stage. So early bloom is always going to be the best time to put a fungicide on, or full bloom is always going to be the best time to put a fungicide on. You really need to look at when is that pathogen coming into the crop, what growth stage the crop is at, and does that coincide with favorable weather conditions. So just to to follow up on that, and we're almost done here, just some thoughts regarding stem rot fungicide timing, because it's a question that we get asked a lot. Which is better? Should I go in at early bloom or wait and go in at full bloom? And it really depends on when the spores of the Sclerotinia fungus are being produced and floating around in the air, potentially infecting the crop. So if you look at the scenario where you've got average rainfall throughout June, so you've had good soil moisture, those sclerotia are going to start the germination process as soon as the crop canopy starts to cover that soil surface and hold the moisture in. So that could be as early as mid-June. So you're going to have production of apothecia as you move into the 3rd and 4th week of June. So as the crop is starting into bolting and into early flower, you already have apothecia present, you have spores in the air, and you have a risk. At that point, I think probably early bloom application may provide some better control. If you look at a scenario where your May has been fairly dry and the first half to 2/3 of June has been fairly dry, so you have conditions that are not conducive for germination and production of apothecia, and so as the crop is coming into early flower, let's say towards the end of June, all of a sudden you start to see moisture. That doesn't automatically translate into spores in the air. It needs about 2 weeks roughly to respond to that moisture. So really those spores are not going to be floating around in the air as the crop's in early bloom, but probably more than likely as the crop is moving into full bloom. So just a couple things here that may help in terms of trying to decide when to spray. And if you've got good moisture conditions in June, good moisture conditions in July, and you've got a yield potential of 50 to 70 bushels per acre, maybe you might want to be looking at considering a split application. I'm not going to touch too much on this. You have the ability to go back, and there's an excellent set of information at NDSU's website illustrating bloom stage, so you can go and look at that at your leisure. But basically, if you look at early bloom, our lower flowers that have been out for a while, we're not seeing a lot of pod growth. But as the crop is moving into full bloom, you start to see, and at 50% flower, usually it's about the time when the crop is at its most intense yellow, you can see some of these lower flowers, the pods are actually starting to elongate. But consult this information, and that'll give you some guidance as far as bloom stage and applying a fungicide. Just very quickly about fungicides, just a bit of a heads up. We have seen some cases of fungicide resistance in Western Canada, and primarily with benomyl, which is the active ingredient in benylate and Sclerotinia, both in canola and alfalfa. And this was work that Bruce Gosen has done, a colleague at Saskatoon. And he was able to find some isolates that were actually quite resistant to benomyl. And it was probably as a consequence of very repeated use of benomyl or benylate in a field under fairly short rotation. So be cognizant of that. If you're using fungicides and you're on a tight rotation, mix things up. Try to avoid using the same product or same group of fungicides between growing seasons. And if you're looking at a split application and fungicide application at early and full bloom, try to go to a completely different fungicide group. And again, this information here, I'm not going to go through it, but there are a number of different fungicides that are currently available, and I apologize if I've missed any, but certainly consult the Provincial Chemical Guidance for more information. Go to this FRAC website, and this is Fungicide Resistance Action Committee, and they have some information as far as fungicides, and basically the risk of developing fungicide resistance that I've indicated comes from this group. And it's a global group comprising not only government pathologists and staff, but it's largely led by industry. So that gives you some of the information as far as some of the different fungicides that are out there, the different groups. So again, if you're looking at tight rotations, even between growing seasons, mix things up. Use a different fungicide group. Don't use the same product if you're in a canola wheat rotation, and especially if you're looking at a split application and a couple applications during the growing season. So I won't say too much about the products. My philosophy as far as comparisons between the different products, they all do well as far as managing Sclerotinia. The key thing is to get them on at the right time and where you've got a reasonable yield potential in terms of that crop responding to that fungicide application. One of the other sort of holy grails, and I won't talk too much about this, this is some work that my colleague Lona Bookvault is working on at Ag Canada in Saskatoon, and Lona most recently has identified some very exciting sources of actual true physiological resistance to Sclerotinia, and actually some pretty impressive levels of resistance to Sclerotinia, and you can see some examples of that here with Westar versus some resistant germplasm from China. So right now, if you look at some of the varieties that are out there and they are referred to as resistance, I would say you're probably looking at reduced susceptibility and not true resistance in the sense of what we're looking at, some of the recent germplasm that Lona's looking at. But certainly with the varieties that are out there that are less susceptible or maybe tolerant, they can provide some benefit as far as managing the risk, but under heavy disease pressure, they're still going to get hit with Sclerotinia, and you may still need to put a fungicide on. So certainly, Lona has got some very interesting germplasm that they've identified here in the last 2 to 3 years, and that is germplasm that's going to be deployed to industry, and then probably you'll see some varieties like probably in that 5 to 10 year time frame. So resistance for Sclerotinia is the holy grail. That, you don't need to worry about making the decision to spray or not to spray if you have true resistance. And we're currently not quite there, but we're certainly moving in that direction, both Ag Canada and provincial and university colleagues, and of course industry. So with that, just to leave you with a bit of philosophy, use an integrated approach to crop and disease management. First and foremost, know what you're dealing with on a field-by-field basis. Review the resource information that you have available. Assess the risk that you have and respond accordingly. So if you need to use a fungicide, use it. If you don't need to use a fungicide and the risk is simply not there, don't use that fungicide. Use it when you actually need to. Optimize crop health, so use good agronomics, good fertility, and you'll promote a healthy crop. Variety choice, use resistant varieties if available, and don't forget seed health. Crop rotation and diversity. Economics come into play here, but as a pathologist, my philosophy is try to extend the rotation as much as possible, and ideally at least 2 years, 2 full years of a non-host crop between host crops, and that will give you some benefit as far as disease management. Now, in terms of integrated disease or crop management, it just isn't the use of a combination of tactics and strategies, so throwing the kitchen sink and everything at managing a pest or a crop issue. It really also involves integrating knowledge regarding the host, the pathogen, and environment. And an example of this with Sclerotinia is the spray timing. So you need to look at when the host is flowering, is the pathogen available, and what are the environmental conditions like? What's the growth stage? Is it early bloom? Is it full bloom? So integrating knowledge as far as the host pathogen and environment. And last but not least, if you look at the benefits of integrated disease, integrated crop management, or in general integrated pest management, look at sustainable management of disease, crop, insect issues, weed issues. You have the ability to deal with multiple issues because often tools that are used for one pest or crop issue are of benefit for other issues. Some of these strategies are actually fairly low input costs, so you can reduce input costs by looking at integrating, using an integrated approach to disease management. Let's take a sip of coffee here. The last 2 points I can't emphasize enough. If you're relying exclusively on either a host resistance or fungicide application as your only disease management or pest management strategy, that's a highly risky situation because these pathogens or weeds or insects can adapt in some cases quite readily to the source of resistance that you have in your variety. or the fungicide that you're using. So if you're using a combination of approaches, you're compensating for things. So you're putting less selection pressure on the pathogen population or weed population or insect population to develop the ability to overcome the host resistance that's in that crop variety you're growing or the fungicide that you're applying. So with that, I'll end there, Derwin, and here's my email address. So I'd be more than happy to try and answer any questions at this time, as well as anything that comes my way via email later on. So thank you for your attention, and I hope you have a great long weekend.
Thank you very much, Kelly. Lots of good information there. I'm just going to bring up the intro slide again. We do have a couple of questions, so I'll pass those along right away. But
Okay.
just before we do that, I'll share. I'm sure lots of the CCAs have been waiting to see the code for accessing their credits. So I've just put that up. It's TURC28 SBC, and if you jot that down now, then when the webinar ends, we'll have a link posted at the Canola Council address for our webinar page, and you can click that link, and it'll take you to a short survey to capture your name and email and your CCA or CCSC number as well as the code. So with that, I'll just share, we do have a couple of questions, and the first one is from Jim, and he's got a good question. Given we've had a fair amount of what the Weather Channel likes to call active weather the last couple of days, I understand, and certainly up in northwest Manitoba, some tornadoes and hail and whatnot, and also throughout Saskatchewan, I understand they've been getting some of that as well. So his question is, would you have any comments in terms of hail damage on the crop at kind of this late rosette to bolting early flowering stage? With that hail damage, which disease would that damage promote infection? Or which disease would you be most concerned about in terms of that damage creating the potential for disease infection? And I guess I'll add the follow-up question of, are you aware of any strategies that growers might use to mitigate that risk if they feel they've still got good yield potential?
I think the main disease that I would be concerned about is blackleg. I've seen over the years, certainly since the early 80s and more recently here in central Alberta, where you've got hail damage, it just provides an avenue of entry for the blackleg fungus. So the key time for infection in canola is prior to the 6th leaf stage, so at a fairly early stage. Later on, the plant tends to become less conducive to infection, so that the stem tissue, you need the infection to be internal by that time. But if you've got hail damage occurring, what that does is it provides a wound site, and we've seen blackleg infections occurring as a consequence of hail damage. Now, at that stage, probably those infections would largely be superficial and likely wouldn't cause huge impacts as far as disease. In terms of something like Sclerotinia, likely the impact would be limited, because if you imagine a wound site on a plant, it's going to be pretty active warfare as far as all the organisms that are floating around in the air trying to take advantage of that wounded tissue. So you're going to have organisms that are typically saprophytes that all of a sudden have the opportunity to start living on that dead tissue, and those things will compete with things like the Sclerotinia fungus or the blackleg fungus. So, you know, in terms of putting on maybe a fungicide in terms of hail damage, really the fungicides are protecting green leaf tissue. That's what your primary focus is with fungicide application. So in the case of blackleg and fungicide application at that stage, again, the infections are largely going to be probably superficial, and likely not going to have a huge impact on yield. So putting a fungicide on is probably not going to have a huge impact. In terms of blackleg, the key time if you're putting a fungicide on to maximize the level of activity is earlier on. So you need to get at those early infections. And that fungicide, in terms of blackleg, you're probably looking at suppression and a reduced level of disease. It's not like what you might encounter with spraying for stripe rust in wheat or some of the cereal leaf diseases. You're getting a reduction in the amount of blackleg, so you might go from 50% infected plants down to 30. So you're still going to have potential yield loss, but the level of yield loss is less. That's I don't know if that addresses the question, but certainly hail damage can provide avenues of entry for some plant diseases. So certainly we've seen it in blackleg, but if you imagine all the other organisms that are there, they're going to invade that plant. So they're primarily affecting the damaged tissue. The healthy, intact tissue that's there, and the new tissue that's produced is not going to be affected by those organisms, especially the saprophytes that are affecting that dead and damaged tissue.
Great, thanks, Kelly. The next question from Ken is, would we be correct in assuming that we're at low risk of Sclerotinia infection if we simply can't find any apothecia in any fields? And his comment was that right now the topsoil surface is fairly dry in their area.
Yeah. If you're in a situation where the soil surface is dry, that's not conducive to germination of the Sclerotia. You really need the soil surface to be wet. So it needs to be close to field capacity to have conditions that favor germination of the Sclerotia and production of apothecia. Once that occurs, if all of a sudden you have maybe several days of dry conditions, those apothecia may wilt a bit, but if you all of a sudden get moisture coming back in, the Sclerotia are already preconditioned. Those apothecia will come back. The Sclerotia will also produce new crops of apothecia, so they can respond quite quickly. But if you're in a field and you have conditions that are, so you've got wet soil conditions, you've got a crop canopy that covers the soil surface, and you can't find apothecia, I would say that don't assume that you don't have a risk. I think if you look at work done by my colleagues in Saskatchewan previously, Richard Google, for instance, and then colleagues in Ontario like Greg Boland at the University of Guelph, and others in New York State and so on, the relationship between apothecia numbers and disease risk is loose, and that's because sometimes these apothecia are produced and their distribution within a field is quite aggregated. So there might have been a lower area in the field where you have production of Sclerotinia in a previous growing season, so that's where most of the apothecia are going to be. And unless you spend several hours out in the field, you may not see that. But you can look at other information that may help to answer the question. So if you don't find apothecia and you've spent a long time doing that, you know that there's limited history of significant levels of Sclerotinia in the field itself and neighboring fields and in the region. In all likelihood, your risk is low. But if you have a field where you've had a really good rotation, very few broadleaf crops, so Macomb, for instance, our beef unit here at the Research Center grows a lot of silage barley, and we've had a lot of trouble getting Sclerotinia going in some of our trials that Randy Kuchner and I have run over the years. It's been extremely frustrating. You might be in a situation like that, but your neighbor's grown a lot of pulse crops or has had canola with Sclerotinia. So if you know that your neighbors or in the area there's been a lot of Sclerotinia in the past few years, significant levels, to me that would indicate that there's a potential risk there, and that even though you're not finding apothecia, there still may be a risk that warrants a fungicide application.
Good.
And that's where the petal testing comes in. That the one advantage of petal testing, and if we get this quantitative PCR working, is that basically you're one step closer to actual disease in the crop. So you're going to account for any spores that are being blown in from adjacent fields, or in the situation where the disease is highly aggregated. So the sclerotia and apothecia are highly aggregated within an individual field.
Right. That's a great point. The next question was with regard to contans, and just what is your opinion on using contans in fields after a susceptible host crop like canola or peas that has had significant Sclerotinia pressure in the previous year? What's your opinion as using contans as a strategy to try and mitigate that risk?
I think contans has been quite effective as far as managing Sclerotinia, but you need to look at how you use it. So if it's the soil incorporated version of it, really what you're doing is helping to speed the decomposition and remove the sclerotia that are within that field from the equation as far as future disease epidemics. So that contans mycopathogen will start to colonize the sclerotia and significantly decrease their viability and ability to survive over a long growing season. So that's within that field itself. The problem is that you may have an inoculum coming in from adjacent fields. So in terms of soil incorporation, it almost has to be sort of a region-wide approach that all fields are treated with it. And initially you may not see a huge reduction, but over time, as you build the levels of the contents in the field, you're going to see a general reduction. Other work that my colleague Debbie McLaren has done has actually shown some benefit as far as spraying content in the crop at flowering and using it as sort of a mycofungicide during the growing season, and that would help to reduce the amount of infection within the crop in a particular growing season. So it depends on what you're targeting. If it's soil incorporated, you're really looking primarily at the overwintering sclerotia within that field, whereas contans at the time of flowering is more preventing disease infection during the growing season. However, I might add that with contans or even fungicide application, anything that reduces the level of disease is going to reduce the amount of sclerotia that are being returned back into that field. And ultimately, that will have an impact in terms of the amount of inoculum that's available. So if you're reducing that through contans sprayed during flowering or using a fungicide, you're going to see a reduction in inoculum levels within that field or area.
Great comments, Kelly. I think we'll do one more question. We've run a little past the hour here, but there is one question that I know comes up fairly often, and the question is, for guys spraying Sclerotinia with a high clearance sprayer versus a plain, his comment is with the high clearance sprayer possibly causing more petal drop or prematurely. Will that affect yield in the end? And I guess it goes back to the pros and cons of higher water volume with the ground rig, but not having the tracks and some of the other issues that go along with the ground rig versus air. So.
I think I wouldn't be as concerned about that, especially if you're using some of the more modern high clearance sprayers. When I was in Saskatchewan working with Robin, he did a lot of work initially comparing aerial versus ground application, and at the time the technology that we were using for ground application was fairly crude. It was the three-wheeled spray coops, which were not very high clearance, and we were looking at other sort of versatile tractor setups with tarps draped along the undercarriage to try and minimize the amount of crop that was damaged. So there was a fair bit of crop damage that occurred with that type of equipment. You see that much less so now with the modern high clearance sprayers. So I wouldn't be as concerned. And the work, the general impression that I think we talked to Robin that we had at the time is that we tended to have better management using ground application with for Sclerotinia. And I know at the time Robin kept encouraging producers to consider tram lines, which was something that's routinely used in Europe and he'd been over in France and Belgium quite a bit, working with colleagues in Sclerotinia there. So it's not something that at the time producers considered, but you know, if you look at the interest in controlled traffic farming nowadays, and the potentially putting in tram lines for all of your field operations, that's something that would help to mitigate that. But you know, I think in the results that we saw back using older ground spray rig technology, we were still seeing a significant benefit as far as putting on fungicide with that type of equipment. And in general, we were seeing better management with the ground rig versus the aerial. However, we still get good results with the aerial too, if it's put on at the right time, at the right rates, and so on.
Great. Well, I think in the interest of time, we'll wrap things up there. Just one other comment. When I put up the code, I also wanted to mention, for anyone that happens to be viewing this as a recording, they've changed the stipulations a little bit in terms of accessing the available credits for viewing a recorded webinar. So we do have someone collecting a short series of questions. So there'll be a quiz, and it will be posted again on our webinar page under the viewer for the webinar, and you'll need to click on that and answer those questions and submit them in order to access the CCA credit for that. And just one final comment that came in that I think I'll pass along. With regard to the contents, I know with all of these biological products, there's lots of research looking at various means of application to try and maximize their effectiveness. But there was just a comment from one of the specialists that the version of contents that's currently registered for use here in Canada, the strategy with it's registered for foliar application, but it's really targeted at that breaking down the sclerotia in the soil, so it's not really registered for an application timing to protect the flowers at this point in time. So just thought I'd pass that along to the groups. There's no confusion about that. But with those and other products, certainly the biologicals, they're looking at different methods of application to try and figure out the best way to apply. So with that, I just want to thank everyone for attending. And I also want to thank Kelly for sharing all of his expertise with us. Lots of good information shared today. And finally, I just want to mention you can go to our webpage to view the webinars at any time. But in terms of promoting future webinars, canolawatch.org is the distribution list that we use a lot for promoting any of our extension activities. So if you're not signed up for that, I'd encourage you to go to the canolawatch.org webpage and sign up. So with that, we'll wrap things up, and thanks very much, Kelly, for presenting today.
Yeah.